Thank you for a wonderful talk. We're now going to move on to the Junior Faculty Research Awardee, and that's Dr. Xin Zhang from Ochsner Medical Center. She's going to talk to us today about small molecule inhibitor selectively reduces T follicular cells and abrogates joint inflammation in a mouse model of rheumatoid arthritis. Dr. Zhang.
Hi, today my talk is a small molecule inhibitor selectively reduce the T follicular helper cell and abrogate joint inflammation in a mouse model of rheumatoid arthritis. I have nothing to declare. Rheumatoid arthritis is a chronic systematic inflammatory disease, affecting about 1% of population. And it's primarily a disease that affects the synovium. And if not treated or uncontrolled, erosion of the cartilage and the bone can develop and resulting in the joint destruction and then affect the quality of the life. The pathogenesis of the rheumatoid arthritis is still not fully understood. It's well known that T cell account for majority of the cell in the rheumatoid arthritis synovium is more than 50%, and the B cell only count less than 5%. And then among the T cell, actually majority of the T cell are Th1 and the Th17 cell. And it's no Th2 and the regular T cell, which play an inhibitory role, is really less, sometimes deficient. It's unlikely that a single rheumatoid antigen exists. A lot of people believe that type 2 collagen play a major role in the development of rheumatoid arthritis. So today I will talk a little bit about the background of T follicular helper cell and then talk about some of the preliminary data we got from the rheumatoid arthritis patient. And then we will, I will focus on the small molecule which targeted in the T follicular helper cell in the mouse model. So T follicular helper cell is a subset of CD4 positive T helper cell. It's usually located in the germinal center of the lymphoid follicle. Usually it's in the spleen, lymph node, or even tonsil. They control the multiple aspects of the B cell immunity, such as selection of the high affinity of the B cell proliferation, and differentiation to the plasma cell. So the T follicular helper cell, it have specific phenotype, which is a CD3 positive, which is T cell marker, and CD4 positive, which is a T helper cell marker. And the specific T follicular helper cell marker is CD57, CXCR5, PD1, and the ICOS. Usually people use the CXCR5 and the ICOS as a typical marker for the identify the T follicular helper cell in patient or in the mice. And then the T follicular helper cell signature cytokine secreted is IL-21, and then secrete a little bit of IL-10, but the majority of IL-21. IL-21 actually play a major role to make the B cell proliferate and differentiate to the plasma cell. And then the T follicular helper cell signature transcription factor is BCL6, which is our small molecule inhibitor targeted. And then this graph actually is published in BioLab in the Ochsner journal. It's a review paper. It's talking about how about T follicular helper cell develop in the second lymphoid organ. So from the left, actually naive T cell express TCR and CCR7 on the surface in the blood, and then they go to the second lymphoid organ, such as the lymph node, and then they interact with dendritic cell, and then became active T cell. After they became active T cell, they gradually increased expression of the ICOS and CXCR5. And then based on the gradient expression of the counterpart of the CXCR5, which is CXCL13, they migrate to the border of the T cell zone and the B cell zone in the germinal center, and then interactive B cell in the borderline. And then after interact with the B cell, like the right square, this part, and then they move to the center of germinal center, move in the center of the germinal center, and then they by secreted the cytokine, if the T cell secreted IL-2 and IL-4, it will make B cell become memory B cell. And then if the T cell secreted more IL-10 and IL-21, the B cell, germinal B cell, will differentiate to the long-lived plasma cell, and which produce the antibody. And then this one is talking about T cell, summarize the T cell subsets, and then the cytokine they produced, and then the function they have. So the similar like last slide, the naive T cell became after interactive with dendritic cell, became active T cell. Induced by different cytokine, they can differentiate to different T cell subsets. And then each different subsets have their unique transcriptional factor, and then have unique surface marker which can identify if you study any T cell subset, you can identify this cell by their surface marker and the transcription factor. And then we focus on the bottom one, T follicular helper cell, which express the CXCR5 and the ICOS, and then secrete the cytokine IL-21, play a role in B cell differentiation and proliferation. And our preliminary data in the rheumatologic arthritis patient, first we check the circulating T follicular helper cell in the RA patient. So the left graph A showed how we define, we gate by the flow cytometry. So by serial gating, CD3 positive, CXCR5 positive, CD20 negative, CD4 positive, and ICOS PD1 double positive. We compared the T follicular helper cell, which is double positive in the ICOS and the PD1, and compared in the healthy group and the RA patient. On the right, you can tell we screened 68 RA patients, and then you can tell in the severe and moderate RA patient, the the T follicular helper cell is significantly increased compared to the healthy control. And then we also checked the T follicular helper cell in the synovial fluid. So we get two synovial fluid from the RA patient. And then for positive control, we use the tonsil and the cut slides for the check their germinal center and then staining T follicular helper cell. You can tell actually this is germinal center in the top row is germinal center in the tonsil, and the bottom is synovial fluid in the RA patient. So we can see actually the existing of the T follicular helper cell in the severe RA patient, which is not exist in the healthy dog. And then we check the frequency of the circulating T follicular helper cell, how they related to the disease severity and then the autoantibody production. Actually, the red one is the close relevant. So the T follicular helper cell percentage actually correlated with the -28, plasma blast, and also anti-CCP, which is a specific autoantibody for the rheumatoid arthritis. But it is not relevant to the R factor and the ESR and the CRP, which is the bottom two is most relevant to the inflammation. So from here we thought T follicular helper cell probably is relevant to the antibody production, which is really makes sense because T follicular helper cell secrete IL-21, which is play a major role in the antibody production. And then after that, we further investigate the role of T follicular helper cell in RA pathogenesis in the mice with the collagen-induced arthritis. So we set up this model just to test to treat the rheumatoid arthritis using the small molecule inhibitor, which is target the transcriptional factor BCL6 of the T follicular helper cell. And then the method is the collagen-induced antigen was induced to the 24 DBA mice by the immunization with the chicken type 2 collagen. And then following the onset of the clinical arthritis, mice were treated with the small molecule inhibitor. And then disease progression was monitored daily and recorded by the swelling of the paw and the ankle from the mice. At the end of the study, the blood, spleen, and affected paws were collected, and then we cut the slice and did the HE staining and the IHC staining for T follicular helper cell. And the mice from the graph A, day 0, you can tell that they have a very beautiful paw for the mice. And then after day 42, after we give the mice the collagen, the paw became swollen. And then the treatment, the mice received treatment, and actually they have a really reduced swollen. So graph B showed how thickness of the pad, and then which showed how swollen of the arthritis, how swollen of the joint, and then the show the severity of the arthritis. So you can tell the disease developed around week. Around day 21, which is day week 3, and then the treatment actually is significantly reduced the swollenness of the paw from the mice. And then we check the T follicular helper cell in the spleen and also the circulating T follicular helper cell. So you can this is significantly actually decreased by the small molecule inhibitor in the spleen. And then in the blood, circulating T follicular helper cell also decreased, but then didn't reach the significance. And then for the IHC staining and the HE staining, you can tell in the spleen for the H and E staining, and then the germinal center is really enlarged in the control rheumatoid arthritis mice, and then treated with the small molecule, the germinal center is kind of like a normal size. And then you can also CXCR5 and staining shows T follicular helper cell, and also the paw. You can see the structure in the paw. Actually, a lot of inflammatory cell deposit in the paw in the control mice, and then the structure actually is deformed. And then the treated with small molecule inhibitor on the T follicular helper cell, the structure actually is very intact, and hardly we can see any T follicular helper cell in the paw. And that's the percentage. We use the imaging scope to count the percentage of the software to count the percentage of the T follicular helper cell. So in the left is the T follicular helper cell in the spleen, and the right is the T follicular helper cell in the paw. So they both actually dramatically increased by the small molecule inhibitor, especially in the paw. So our conclusion is the T follicular helper cell is originated from the spleen lymphoid follicle, may contribute to the circulating T follicular helper cell in the peripheral blood in the our rheumatoid arthritis mice model, and play an important role in the development of active rheumatoid arthritis. The small molecule inhibitor targeted to the BCL6 can lead to a decrease in the T follicular helper cell in the spleen, and then inflame the joint in the CIA mice model. It could be a new additional therapy for the rheumatoid arthritis in the patient. Thank you.